Stem cells are one of the major discoveries of the 21st century. In fact, a large number of researchers swear by them. The unique characteristics of stem cells enable scientists to use them to create any type of human cell, forcing them to express certain genes, as in this case.
The stem cells are the cells that do specifically this. They perform the function of producing new and mature cells that go into the tissue to replace those that have been lost to injury or simply consumed by the physiological function of the tissue. That's why a stem cell is so important. Without a stem cell, your body cannot survive. It will essentially degrade itself and you will die.
Before being able to use nerve stem cells to treat certain diseases of the brain, scientists need to be able to master the genes that control them. This is where the motivation to create the Plurigenes project came about. This network brings together 9 European laboratories. Their aim is to identify the genes that control the state of stem cells found in the nervous system. Here, in this French CNRS laboratory, tropical fish are used to investigate these mechanisms.
We use model animals that are small fish because they have many more cell population that proliferate at adulthood than us. We have only 2 populations of neural stem cells in our brain. So those fish have many more population of cells that divide, and they have many more neural stem cell population.
These fish have been genetically modified to make certain parts fluorescent. This enables the behavior of certain fundamental genes shared with humans to be studied. By inhibiting the action of these genes, scientists could make some of our body's cells revert to the state of embryonic stem cells. This revolutionary technique would open the door to a large number of future therapeutic uses.
Plurigenes try to identify the genetic elements, the genes, that are involved in the process of de-differentiation, going from a mature adult cell to a more juvenile state. That cell will come from your skin cell and will be reprogrammed to make any kind of cells of your body. If you were to use those cells for transplantation in your body, those are your cells. There is no rejection. In other words, you have cloned embryonic stem cells, but you have never cloned an embryo. You have no rejection. You have a very proficient, optimal, I would say, solution to do transplantation, cell therapy, whatever you like to use the cells for, without any ethical issue and without any rejection.
Thanks to the advances made in fundamental research under the Plurigenes project, clinical trials may begin. They will start in Italy in a few months' time, working on patients with presently incurable brain diseases.